Good afternoon. I am Dr. Kara Wilder, a technical writer at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Reclassification of the Mycobacterium tuberculosis complex species as Mycobacterium tuberculosis, presented by Dr. Marco Riojas. Dr. Riojas is a scientist at ATCC and BEI Resources. He has a PhD in biodefense and his primary research interests include biodefense, bacterial systematics, phylogenomics, and genomic identification of species. In the following webinar, he will discuss the techniques used to examine the Mycobacterium tuberculosis complex and how the results from the study affected the reclassification of the species within the complex. If you have any questions for Dr. Riojas, please use the questions box available through the webinar program. Questions will be answered as time allows and will be archived with a recorded webinar presentation on the ATCC website at www.atcc.org.
Thank you, Kara. This webinar will focus on a paper that ATCC recently published in the International Journal of Systematic and Evolutionary Microbiology, or IJSEM. The title of our paper is Phylogenomic Analysis of the Species of the Mycobacterium tuberculosis complex demonstrates that Mycobacterium africanum, Mycobacterium bovis, Mycobacterium caprae, Mycobacterium microti, and Mycobacterium pinnipedii are later heterotypic synonyms of Mycobacterium tuberculosis. It can be found in the January 2018 issue of IJSEM. This paper showed that what were previously considered separate species within the Mycobacterium tuberculosis complex are actually a single species, namely Mycobacterium tuberculosis. As a brief outline for this webinar, I'll first describe ATCC and our mission. In order to properly discuss our paper, we'll need to cover some of the technical background of bacterial systematics, so I'll describe some concepts relating to bacterial species, how they are assigned, and more modern identification techniques, including DNA Dna hybridization and digital DNA Dna hybridization. Finally, I'll discuss our paper, including our methodology, results, and conclusions, and the nomenclatural proposal made to IJSEM. First, here's a brief introduction on ATCC. ATCC was founded in 1925 and is a private, independent, nonprofit organization headquartered in Manassas, Virginia. We also have an R&D facility in Gaithersburg, Maryland. ATCC is the world's premier biological materials resource and standards development organization. We collaborate with and support the scientific community with industry-standard products and innovative solutions. Our broad range of products include cell lines, microorganisms, genomic and synthetic nucleic acids, and media and reagents. Each of our biomaterials are expertly authenticated and characterized using genotypic, phenotypic, and functional analyses. Now let's take a look at what a bacterial species is and how bacterial species are determined. Every bacterial species is represented by a specific strain that is known as a nomenclatural type strain. The publication describing the species also includes a detailed description of its characteristics. These characteristics at a minimum include cellular morphology, results of biochemical and metabolic assays, and culture conditions. More recent descriptions often include mass spectrometry data, 16S ribosomal RNA gene sequence, and even whole genome sequence. Because the publication describing a new species is generally published shortly after its discovery, the type strain of a species is usually the first one that was identified. However, this isn't always the case. In cases where an atypical isolate has been designated as the type strain, the characteristics of the type strain may not be completely aligned with those of the species in every respect. Certain biochemical results or other characteristics may differ within the typically expected variation of such characteristics. However, the overall characteristics of a type strain are generally representative of the species as a whole. Type strains are designated with an uppercase T as a superscript after the strain name. The example most relevant to this presentation is the H37RV strain of Mycobacterium tuberculosis, which is shown here in the proper format indicating it as a type strain. Because the type strain of a species is an officially recognized entity describing the species, it can essentially be thought of as forming the definition of the species. When a new strain of bacteria is isolated, the bacterial type strains serve as comparative references to identify the new strain and assign it to existing species. Alternatively, if the new strain does not match closely enough to any existing type strains, it is these comparisons that help exclude the new strain as an existing species and allow it to be proposed as a novel species. This is accomplished by comparing the properties observed in the new isolate to the characteristics of the type strains. Historically, this has been done by comparing the phenotypic characteristics of bacterial strains. A strain that shows enough similarity of the essential characteristics of a type strain is said to be within the circumscription of that type strain. Because it is circumscribed by that type strain, it therefore belongs to the species. As a very oversimplified example, let us imagine that we have 4 type strains of 4 different species. Species A, B, C, and D. Each of these vary in some combination across 4 different characteristics: gram stain, atmospheric tolerance, motility, and nitrate reduction. If we isolate a new strain of bacteria, we can examine its characteristics and compare them against those of the type strains. In this case, our new bacterium is gram-negative, aerobic, motile, and nitrate positive. Based on the comparisons to the type strain, we can clearly see that its characteristics most closely align with those of species C. Thus, we can assign it to species C and say it is a strain of that species. Of course, we recognize today that many phenotypic characteristics can be unreliable. This can be particularly true for biochemical assays. In the example on the previous slide, we used the nitrate reduction activity of the new strain of bacteria as one of the criteria to help identify it. Because species C is nitrate positive and our new strain is nitrate positive, we were able to ID it as a strain of species C. This is because the new strain presumably has the same biochemical pathway for nitrate reduction as the type strain. However, it could have an entirely different biochemical pathway that also happens to reduce nitrate. Or in an example that would be even more confounding to our interpretation, it could have the same biochemical pathway as the type strain, but one of the genes in the pathway could have a mutation that inactivates a critical protein in the pathway. This inactivation could result in a lack of nitrate reduction, even though the biochemical pathway is actually there, albeit in a mutated and inactive state. If comparing this new nitrate-negative strain to the type strains we saw previously, it might be more tempting to categorize the new strain with a type strain that lacks a nitrate reduction pathway just because the observational endpoint for both is similar. This, of course, would be incorrect. Of course, this example is oversimplified in both the number of characteristics that are considered as well as the over-reliance on a single characteristic. In reality, such comparisons would be made upon a battery of biochemical tests and other criteria. However, even in combination with many such assays, identification solely by phenotype can be unreliable. Because of this, we rely more heavily on genotypic comparisons today. Typically, the first choice for a genomic comparison is the 16S ribosomal RNA gene. The 16S gene contains some hypervariable regions that generally vary between bacterial species, flanked by some regions that are very highly conserved across all bacterial species. These conserved regions can be used for universal amplification of the gene, and DNA sequencing can be used to determine the nucleotide sequence of the gene. It can then be compared to the 16S sequences of existing species and either identified or possibly established as a new species. Of course, it's not always as straightforward as this. The 16S gene is not always differential between different species. This is particularly true in some well-known examples, such as the Bacillus cereus group, as well as some mycobacterial species, including the Mycobacterium tuberculosis complex. In cases such as these, we can expand our genomic comparison to additional genes. Two commonly used genes include the housekeeping genes HSP65 and ARPOB. Another approach is to expand the comparison to a number of different genetic loci using a technique called multi-locus sequence typing. However, even these approaches can still provide misleading results. In particular, these techniques can be vulnerable to selection bias. In picking the gene or genes to use for comparison, you might be choosing the one gene where very similar bacteria just happen to be different, or you might be choosing the one gene where very different bacteria just happen to be similar. Naturally, increasing the number of genes used in the analysis reduces this risk. By extension, the most accurate genetic comparison would be the one that encompasses the entire genome. This would, by definition, include all the genetic information contained within an organism, i.e., everything that makes that organism what it is. And because the whole genome would be used for comparison, it should be entirely objective and not vulnerable to the same sort of selection bias as choosing specific genes. One method to compare the whole genomes of two organisms is called Dna Dna Hybridization, or DDH. In this wet lab technique that dates back to the 1970s, the genomes of two different organisms, let's call them A and B, are isolated. Each of the genomes is denatured, then allowed to anneal together in a hybrid assembly containing one strand of organism A and one strand of organism B. The hybrid strands are then heated to denature them again. The amount of energy required to separate the strands is proportional to the number of hydrogen bonds formed between the strands, i.e., the sequence similarity between the genomes of the two organisms. The DDH threshold for species delineation is 70%. So two genomes with a greater than 70% DDH similarity are the same species as each other. Conversely, two genomes with a less than 70% DDH similarity belong to two different species. It's important, however, to emphasize that DDH may tell you that the two genomes are from the same species, but it doesn't tell you what that species is. For example, let's say that genome A is actually from Mycobacterium tuberculosis, but is incorrectly labeled as being from E. coli. When you extract your genome B DNA from another Mycobacterium tuberculosis strain and compare it to genome A, DDH will tell you that genome B is the same species as genome A, which is correct, but you will reach the wrong conclusion because you think that species you're comparing it against is E. coli when it's not. When seeking to use this method for species identification, this is why it is so important to base such comparisons on the species type strains rather than on non-type strain isolates. Because the comparison is being made against the definition of the species, it is by definition correct. DDH is considered the gold standard for species definition. In order to be recognized as a new species, the strain being proposed as a new species must be shown to have less than 70% DDH similarity to the type strains of existing species. However, DDH has some very significant challenges. Namely, this wet lab technique is very labor-intensive, time-consuming, error-prone, and poorly reproducible. Obviously, these are undesirable characteristics of any scientific assay. To overcome these challenges, a sequence-based technique has been developed to be analogous to DDH. This technique is called digital DNA Dna Hybridization, or DDDH. DDDH compares the whole genome sequence from two strains against each other and uses a bioinformatic algorithm to infer their distance from each other. DDDH can be calculated using the genome-to-genome distance calculator, or GGDC. GGDC is an excellent tool that was developed by the German culture collection DSMZ and is available from their web server. Unlike other tools such as average nucleotide identity, DDDH has been calibrated to the same threshold as the gold standard DDH. In addition to assignment of species, DDDH can also be used for assignment of subspecies. The subspecies delineation threshold has been proposed at between 79 and 80%. For the purposes of our research, we used 80% as a threshold. Using these thresholds, we have the DDDH scheme used in our research. In this scheme, a DDDH value above 80%, shown in green, means that two strains are both from the same species and subspecies. Alternatively, if a species has not been divided into subspecies, this would indicate that the two strains are simply the same species. A DDDH value between 70 and 80%, shown in yellow, indicates that the two strains are from different subspecies of the same species. By extension, if a species has not yet been divided into subspecies, this would indicate that the new strain represents a novel subspecies of the existing species. And a value lower than 70%, shown in red, means that the two strains are from entirely different species. Sometimes the genomic data can provide a more modern look at older historical strains. A strain that was once believed to be one species may turn out to be a different species. Let's look back at our simple example from earlier, where we have 4 type strains and 1 isolate that we identified as species C. From the phenotypic characteristics we observed, that identification appears to be correct, but we can enhance our investigation by using DDDH to examine the genomes of these strains. So we extract DNA from each of our species type strains and sequence it to use as our reference for comparison. We then extract DNA from our isolate and sequence that, and we might find that at the genomic level, it is actually different enough to be considered a different species, despite the phenotypic similarities. By comparing our isolate's genome against another type strain genome, we may be able to identify the strain to which it does match, in this case species E. We can then say that based on the more detailed whole genome sequence, the isolate in question doesn't actually belong to species C like we originally thought, based on our more limited phenotypic observations, but is instead a strain of species E. So now let's take a look at the Mycobacterium tuberculosis complex. The Mycobacterium tuberculosis complex, or MTBC, is made up of 6 officially recognized species. These species are Mycobacterium tuberculosis, Mycobacterium africanum, Mycobacterium bovis, Mycobacterium caprae, Mycobacterium microti, and Mycobacterium pinnipedii. In addition, there are a few additional species that are sometimes referred to but are not officially recognized. These include Mycobacterium canettii, Mycobacterium mungi, and Mycobacterium orygis. All of these species are very closely related to each other. They essentially all cause the respiratory disease tuberculosis in different animal hosts, including humans. Some of these are better adapted to certain hosts than to others. For example, Mycobacterium pinnipedii was isolated from marine mammals. The most common MTBC infections in humans are caused by Mycobacterium tuberculosis, Mycobacterium africanum, Mycobacterium bovis, and Mycobacterium canettii. However, most, if not all, of the MTBC species are capable of causing some level of human disease, particularly among immunocompromised individuals. It has been known for a long time that the species of the MTBC are very closely related, and it has been hypothesized that they are in fact a single species. Some recent research investigated the relationship between some of the MTBC species. By comparing various strains of Mycobacterium tuberculosis, Mycobacterium africanum, Mycobacterium bovis, and Mycobacterium canettii to the type strain of Mycobacterium tuberculosis, this research found that all of the strains they examined fall within the circumscription of Mycobacterium tuberculosis H37RV. The conclusion that this paper reached was that mycobacterial scientists should agree in accord that designates MTBC as a single species in the official taxonomic rules of nomenclature. The research findings described in this paper are sound, but their conclusion is overstated. Because the strains they examined were non-type strains, the results that they are circumscribed by Mycobacterium tuberculosis extend only to those particular strains, not to their species as a whole. For example, it can be said that Mycobacterium africanum, strain GM041182, is actually a strain of Mycobacterium tuberculosis. However, although the paper's conclusion seems to indicate this, it cannot be concluded from their work that all strains of Mycobacterium africanum are actually strains of Mycobacterium tuberculosis. Such a more wide-ranging conclusion can only be reached by showing that the type strain of Mycobacterium africanum, i.e. the definition itself of Mycobacterium africanum, falls within the circumscription of MTB. So this is precisely what we sought to do in our research. Of the 6 officially recognized species, only Mycobacterium tuberculosis had whole genome sequence available for its type strain. The type strains of M. africanum, bovis, caprae, microti, and pinnipedii had not yet been sequenced. So in our work, we extracted genomic DNA from the type strains of the 5 missing species, shown here in Table 1 from our paper. We sequenced these genomes using Illumina sequencing and compared these whole genome sequences to each other, as well as to the genomes of H37RV, 3 non-validly published species, and 2 outgroups. We performed these comparisons via both DDDH using the GGDC and average nucleotide identity using ortho-ANI. So now let's take a look at our results. The primary results of our research are shown in Table 2 from our paper. This table shows the results of pairwise comparisons between 9 species from the MTBC and 2 outgroups against the same set of samples. The diagonal line down the middle is the 100% self-comparison line that you would expect from comparing each of the genomes against itself. Above this diagonal, we show the results of the pairwise DDDH comparisons, while below the diagonal, we show the results of the pairwise ANI comparisons. Each cell within the table is color-coded to illustrate the conclusion that can be reached regarding whether it is the same species or a different species. For example, the cell comparing Mycobacterium bovis to Mycobacterium microti via DDDH shows a 98.2% DDDH similarity. This is well above the 70% threshold that would indicate they are different species. It is even far above the 70 to 80% range that would indicate that they are different subspecies of the same species. The same is true of the ANI results, which show an average nucleotide identity of 99.79%. Again, this is well above the 96% threshold that would indicate that these are different species. In fact, looking at the entirety of the table, it becomes clear that all of the species of the MTBC are extremely closely related. The type strains of all of these species are circumscribed by each other, including, most importantly, by H37RV. The reason that this is particularly important is that in cases where multiple strains are found to actually represent the same species, the rules of nomenclature state that the species which was validly published first takes priority. In this case, Mycobacterium tuberculosis was published first and indicates that all of these strains are circumscribed by MTB. Mycobacterium tuberculosis therefore has nomenclatural priority over all the other names. In Figure 1 from our paper, we show a phylogenomic tree generated from the DDDH analysis of the MTBC species, the 2 outgroups, and the type strains of a variety of other Mycobacterium species. The extremely short genetic distances that separate the members of the MTBC contrast greatly with the overall genetic distances shown in the remainder of the tree. This further supports the hypothesis that the MTBC is closely enough related to be considered a single species. In addition to the type strains of the MTBC species, we also examined all the MTBC strains with draft or complete whole genomes in GenBank and used DDDH to compare those strains to Mycobacterium tuberculosis H37RV. We examined 30 strains of Mycobacterium africanum, 69 strains of Mycobacterium bovis, 9 strains of Mycobacterium canettii, 2 strains of M. caprae, 1 strain of M. microti, and 3,631 strains of Mycobacterium tuberculosis. The results of this are shown in summary form in Table 3 from our paper. As you can see, all of the strains we examined showed a DDDH value greater than the 70% delineation threshold and even greater than the 80% subspecies delineation threshold. This indicates that all of the strains with genomes in GenBank fall entirely within the circumscription of Mycobacterium tuberculosis and aren't even different enough to be considered different subspecies. The complete list of results for the GenBank comparisons is available as supplementary material on the IJSCM website. You'll note, however, that there was one strain that was excluded from this analysis. When compared to H37RV, the genome for this strain showed a DDDH value of 22.2% and an ANI value of 79.39%, indicating quite clearly that it is not Mycobacterium tuberculosis. So we examined this genome more closely by using DDDH to compare this strain to the type strains of all the other Mycobacterium species. Using this approach, we were able to identify that the genome deposited into GenBank as M. tuberculosis strain TKK010051 is in fact a strain of Mycobacterium columbiense that was deposited with an incorrect name. The image shown here is a partial look at those results. The complete table of results is available as supplementary material on the IJSCM website. Finally, we reach the conclusion of our research. Since we found that the type strains of all the MTBC species fall within the circumscription of Mycobacterium tuberculosis H37RV, and M. tuberculosis carries nomenclatural priority over all the other names, we have submitted a paper describing a unification of the MTBC species as Mycobacterium tuberculosis. This paper was published in IJSCM Early Access Online in December 2017 and officially in the January 2018 issue. The groups that were previously known as the MTBC species should now instead be considered intra-subspecific subdivisions, i.e. variants, of M. tuberculosis. The term infra-subspecific indicates that this subdivision is even lower than the subspecies level, as none of the strains we examined were different enough to even be considered subspecies. We recommend the use of the former species name as the variant designation, for example, Mycobacterium tuberculosis variant bovis. Table 4 from our paper shows the previous names of the species and the name we have recommended. Because a species can only have one type strain, the type strain of the newly unified Mycobacterium tuberculosis remains H37RV. The type strains of what were previously known as the other species are now considered reference strains, i.e. non-type strains, of Mycobacterium tuberculosis. In the table shown here, you can see that these variant strain names no longer have the superscript T associated with them. So, in conclusion, we phylogenomically compared the type strains of all the species within the Mycobacterium tuberculosis complex and found that they are in fact a single species. In accordance with the rules of bacterial nomenclature, we have proposed that all of these species be considered as strains of Mycobacterium tuberculosis rather than as separate species. We also used digital DNA Dna hybridization to compare a strain ostensibly belonging to Mycobacterium tuberculosis against all the other type strains of Mycobacterium species and discover its true identity. Using this approach, we have shown that whole genome sequencing of bacterial type strains enables the elucidation of the true relationships between species and strains. This has allowed us to modernize taxonomic relationships using the complete genetic information contained within these organisms, including the development of a detailed phylogenomic tree showing the position of the MTBC members relative to the other species of Mycobacterium, which supports our conclusion that these represent a single species. And here are the references for some of the information contained in this presentation, including the full reference for our paper.
Thank you, Dr. Riojas, for that excellent presentation. In just a few moments, we will begin our question and answer session. Please use the questions box available through the webinar program to submit questions. The session will be documented and archived along with the recorded webinar presentation on the ATCC website at www.atcc.org/webinars. Our first question is, does publication of this paper mean that the scientific community has to use the names you describe? For example, Mycobacterium tuberculosis variant africanum.
That's an excellent question, and an important one to make clear. So the short answer to that question is no, that is not required. The longer answer is actually still no, but it's good to consider the two interlinked main components of bacterial systematics separately. Bacterial systematics is the system of classifying bacteria and assigning them names, and this breaks down into two main components. The first is taxonomy, which is a study of organizing different items, in this case bacteria, into various groups based on similarities and differences. The second is nomenclature, which is the assignment of correct scientific names according to standard conventions. Taxonomy is what actually classifies bacteria as different or the same and establishes their relationships to each other. Nomenclature is the names we give to those groups to indicate that they are either similar or different. Of course, the reason I said that these are interlinked is because we first use taxonomy to define taxonomic groups, then nomenclature to give those groups names. So they're not entirely independent. So first let's take a look at taxonomy. Taxonomy is to some degree a matter of scientific opinion. When scientific evidence is published showing a different taxonomy than was previously believed, not everyone is immediately accepting of it. Some scientists may disagree with the new taxonomic conclusions, often based on their own previous research or the perceived shortcomings of the new research. For nomenclature, IJSCM is essentially the arbiter and gatekeeper of bacterial nomenclature. Publication of a name in IJSCM signifies approval of that particular nomenclature. So any name that has been published in IJSCM is considered valid. What that means is that as taxonomy becomes updated, several different equally valid names may be used according to the scientific opinion of the individual researchers. So a good example of this, and one that we mention in our paper, is what we call in our paper Mycobacterium tuberculosis variant caprae. When it was first isolated from goats, it was given the name Mycobacterium tuberculosis subspecies caprae. Then it was later found to be more closely related to Mycobacterium bovis, and it was given the name Mycobacterium bovis subspecies caprae. Then it was found to be different enough from Mycobacterium bovis to warrant being its own unique species, and it was given the name Mycobacterium caprae. What our research shows is that in fact none of the MTBC, including Mycobacterium caprae, are different enough from Mycobacterium tuberculosis to be considered as different species. So our nomenclatural conclusion is that it should simply be named Mycobacterium tuberculosis, specifically Mycobacterium tuberculosis variant caprae. And again, we use that variant term rather than the original term subspecies because we found that they aren't even different enough to be considered subspecies. So to answer the question, if you scientifically agree with our conclusions, you should refer to this organism as Mycobacterium tuberculosis variant caprae. If you disagree with our conclusions, you should refer to this organism as the most appropriate, validly published name that aligns with your taxonomic opinion, most likely Mycobacterium caprae. So the ultimate answer to the question is simply that what you and the scientific community call these organisms is up to you and your individual scientific opinion. Obviously, we think that the variant-based scheme that we proposed is best, since it's based on a comparison of the whole genome sequences of all the type strains. But as long as you use a name that is validly published, it isn't wrong.
Our next question is, if the Mycobacterium tuberculosis complex is actually a single species as your research shows, how is it that these were initially incorrectly identified as different species?
That's another really good question. It's not entirely fair to say that they were incorrectly identified. It's important to think about these identifications in the context of scientific and technological history. Mycobacterium tuberculosis dates back to the late 1800s. M. bovis was originally identified at the beginning of the 1900s, and M. africanum was identified as its own species in the 1960s. Technology has clearly improved a lot since those days, and while we initially had to rely only on gross morphology as the basis for our taxonomic classification, we then moved to biochemical characterizations, and then eventually to the early genetic tests like 16S. So as technology has advanced, we've been able to take closer and more accurate looks at organisms and update our taxonomy accordingly. So the previous taxonomic groups weren't so much incorrectly identified, but they were identified within the limitations of the technology of the time. The most modern technology at this time is whole genome sequencing, which allows us to use the entirety of an organism's genome to make these taxonomic decisions, and that's why we chose to use this in our research. And yes, it is possible that one day newer technology or taxonomic criteria may come along that overrides our nomenclatural scheme as well.
Our next question is, what are the repercussions of the new taxonomic changes for clinicians?
For clinicians or laboratory workers, this change should have no effect. A patient infected with any Mycobacterium tuberculosis variant requires the same treatment and precautions during management. The only thing that has changed is the nomenclature.
Does this mean that there are really no differences between these different taxa that were previously known as species?
Definitely not. And we made a point in our paper to emphasize that our work does not imply that there are no differences. Even in the previous species-based taxonomic scheme, some of the species could further be subdivided into lineages that showed specific characteristics that were stable and differential. For example, Mycobacterium africanum showed a very distinct differentiation into two West African lineages based on some regions of differences and spoligotyping. These different lineages still exist as stable and coherent entities, and we aren't claiming otherwise. We're simply saying that these differences between the lineages don't rise to the level of different species, or for that matter, even different subspecies.
So we're going to take one last question, and that is this type of analysis specific to Mycobacterium, or can it be applied more broadly to other species?
Another great question. It's not specific to Mycobacterium, and it can absolutely be applied to other species. If you recall during the description of DDH and DDDH, I described how it can be used to tell whether two genomes belong to the same species or not. That can be any species at all. The sort of analysis that we reported in this paper is to compare a particular isolate against a species type strain to determine whether they belong to the same species, independent of what that species is. There is one slight limitation to this type of analysis, and that is that the whole genome sequence of the type strain must be available to compare to. But as long as that type strain, for example E. coli, is available, you can take the whole genome sequence of any isolate and determine whether it falls within the circumscription of the type strain of E. coli. Here at ATCC, we're beginning to use this type of whole genome analysis to identify some of our organisms, and this allows us to apply the most modern whole genome methods to the characterization and authentication of our material.
So at this time, we will conclude our question and answer session. I would like to thank Dr. Riojas for the excellent presentation, and thank you everyone for attending the webinar. Any questions that were not answered this afternoon will be answered and posted with a recording at www.atcc.org/webinars. Also, please join us for more webinars in the ATCC Excellence in Research webinar series. You can register for more of our upcoming webinars at www.atcc.org/webinars. For more information about this taxonomic change, please take a look at the publication by Riojas et al. In the International Journal of Systematic and Evolutionary Microbiology. Thank you again, and have a great day.